Okay, well, welcome everybody to Ward Laboratories webinar on what are our soil health analysis telling us? What are our tests telling us? Some questions about soil health. I've got a tremendous group of speakers here. We're going to give it just a few minutes to get things rolling so that we can make sure everybody's on and that everybody can have a chance to join and kind of get settled in. So give us just a minute. You'll see some of our speakers are here and ready to go. I am presenting live from my office, so enjoy some of the artwork in the background here. It's certainly some of the best decoration I can have. So stay tuned. Again, I'm Ryan Dennhardt with Ward Laboratories, and we are coming to you live. We're going to break down our soil health tests, what kind of questions you have, and we've got a great panel of speakers here, and just really to give folks an opportunity to ask some great questions and really have a better understanding. So we'll give it just a few more minutes to let people get in and get settled, and then we'll get going.
In the meantime, while people are getting settled, how about you put something in the chat and tell us where you're calling in from? So are you in Indiana? Are you in Western Australia? I see there's probably people registering from all over the place. So put in your chat in that box. Santa Cruz.
Wow, we got Columbus, Ohio, Santa Cruz, California. We got
Australia.
Australia, wow. Wow, Nicole, you really draw a crowd here, it would appear.
Good morning.
Southeast Iowa, fantastic. Well, as folks are starting to roll in, we'll go ahead and get things started here. Let me mute out some of the other things I've got going on in my office as we continue to get things rolling. So again, guys, thank you so much for joining us. We're excited to be here. We've been talking a lot about this, and one of the things that we hear all the time as we travel and we meet with our customers and meet with folks within regenerative agriculture and just different opportunities that people are like, hey, you know, what's our soil health test telling us? What is the data? What am I looking at? And how can we make that make sense for us in such a way that we can turn that into valuable information to truly turn it into practical application within our operation. So we thought, what better time than now to really get some of the best in the business in here to chat through some of that with you. So I'm going to go ahead and kick it off and introduce some of our speakers as we get going. So I'll go ahead and start with Alexis Hobbs. So Alexis Hobbs joined Ward Laboratories in 2019. She graduated from the University of Nebraska at Kearney in 2017 with a bachelor's in molecular bachelor's in molecular biology, and then she graduated with her master's in biology as well. Alexis isn't just from the website. She's here in person in the lab. She works side by side with the folks running the tests. She is our soil health coordinator, and so we have broadened Alexis's role to really just have her hands in every process of our soil health department. So Alexis, why don't you tell us just a little bit about what you're working on currently?
So currently, I'm really excited about our compost tea testing. So we recently started compost tea testing with the phospholipid fatty acid analysis. So that was really exciting to get off the ground and get going. So if you are interested in compost teas, we've got you covered.
Thanks, Alexis. Yeah, compost tea, that's a big deal. So we're really excited about that, and we're even more excited to have you working on that for us, Alexis. So thanks for that. Moving on, we are also joined by Willie Pretorius. Willie is renowned in the soil health industry, and we are lucky enough to have him on our full-time staff at Ward Laboratories as a soil health consultant for us. Willie, I would love to try to talk through your bio as far as your education and some of these things, but it's pretty impressive. So it looks like you majored in fruit science, horticulture, and plant physiology. You also have some business, some degree in business, and of course agricultural research, managing food companies and soil health. Hailing from South Africa, Willie, why don't you tell us just a little bit about what you're currently working on here? I mean, if you can put it all into a short snippet, I know we've got you doing a lot of things behind the scenes for us. So welcome, Willie, and tell us a little bit about what you're working on.
Hey, thanks, Ryan. Yeah, I have a background in plant physiology, plant biochemistry, and food science, and spent the last 14 years just updating myself on the new science of soil health, and I had to do that all by myself because nowhere could you log onto a website or an opportunity where you could actually learn the science. You had to pick it up piece by piece, and in doing so, I tried to establish relationships with the most qualified scientists and farmers in the world. And I'm glad to say that I have a resource base of extremely well informed scientists in the soil health and regenerative ag world, and as well as probably some of the best icon farmers in the industry. And really, I keep myself busy talking to them and updating myself and putting presentations together. So yeah, I'll be happy to answer any questions that I have the ability to answer, and I'm extremely happy to be on the ward staff, and trust that I can make a small contribution in this field.
Well, I'll tell you, we feel like we've got a stacked, with football season, in full swing now, we feel like we've got a stacked roster with Willie and Alexis handling our soil health operations here. So we're extremely excited to have you, Willie, and I know you're going to be an amazing complement to Alexis. So glad to have you on this webinar, but more importantly, glad to have you on the team. We do have one question before I introduce Nicole. This is part 2, and yes, we can send out a link to part 1, Rhonda. So if you just give us a little bit, I can get you that information. So to answer the first question, we will send out a link to part 1. And without further ado, we were extremely excited. This is our second opportunity to work in this environment with our next panelist. We've done business with her for some time now and have been running samples and working closely in that department. But we are just extremely excited to have Nicole Masters join us and kind of linking arms and helping to educate and bring information in such a platform that people can reach it, as I know that not everybody can travel and get out in person. So we're just extremely excited to have Nicole Masters. So I'll give you a little bit of background about her. Nicole Masters is an independent agriologist, systems thinker, author, and educator. She is recognized as a knowledgeable and dynamic speaker on the topic of soil health. Her team of soil coaches at Integrity Soils work alongside producers in the US, Canada, and across Australasia, supporting producers. Thank you. Supporting producers who work with 1.2 million acres to take their operation to the next level in nutrient density, profitability, and environmental outcomes. She is one of a growing number of people who are facilitating a rapidly expanding world of quality food production and biological economies. Her book, titled For the Love of Soil, showcases examples of the tools and principles producers are using to regenerate their soils, describing a step-by-step triage of actions so that you too can regenerate your land. Website to find that is www.integritysoils.co.nz. So without further ado, welcome Nicole Masters and tell us a little bit about what you're working on.
Thanks for having me, Ryan, and to the ward team. It's a real honor to be here. Great introduction. Thank you very much. It's good to plug the book. Yeah, and I think right now, COVID has given me that opportunity to kind of reassess and draw inwards. So when COVID hit, I canceled 44, I think we're up to 46 full day workshop events. And so it really got me rethinking, what's important in life and how do we communicate in this time where, as someone that does workshops all the time with lots of people, how do we still continue to get that educational message out? So I've just finished a online course. So it's a soil health foundation program, and it's really looking at how do we assess soil health from not only from soil testing and plant tissue testing, but like using all of our senses and doing infiltration tests, or making a Burley's funnel for yourself so you can look at your insect counts and stuff like that. So I've been working on that, and then also at the same time learning to just balance my life a bit more, because I was just, I think a lot of people in this field, you know, you feel like there's a sense of urgency. I mean, sitting right now, I can't see any of the mountains around me. We're sitting in dense smoke, and I'm in Montana, you know, and we're dealing with smoke from off on the West Coast, and it's like, it's easy to feel like we're just running all the time. And so I'm really learning to spend more time with my dog and my horse, and you know, being in observation and being in nature. And I think that's what I'm really loving at the moment.
And that's fantastic. You know, I think there's probably plenty of people that could say they would rather spend time with their dog than anybody else. I know around here, you know, we're pretty dog friendly. So, well, you know what? Without further ado, I'll give you a couple housekeeping things. So, folks, if you have questions, we want to make sure we get those answered. So please participate in the Q&A section on the Zoom platform. If you put it in there, we'll make sure that the questions get asked and answered appropriately. And if we don't necessarily get to all of your questions, we will make sure that we can send out answers and get you guys all set up and ready to go. So we've got some questions already. So we'll kind of start with a basic question. I think everybody would agree is certainly a good place to start. So what is the difference between soil health tests like the Haney test and the standard chemical soil test? What's the difference in soil health tests like that?
So I can go ahead and take this one. So the difference really in the Haney test is going to be your nitrogen value. So standard chemical tests are going to test for only nitrate and ammonia. The Haney test really tests for nitrate, ammonia, and the organic nitrogen, which is really important. You're really getting the whole picture of what's going on in your soil. Whereas with the nitrate and ammonia, you're really only getting half of your nitrogen picture. So you can really, in theory, you can really cut down your inputs by quite a bit by actually testing for the organic nitrogen portion of your soil.
Okay, that's great. Nicole, Willie, anything else to add to that?
Yeah, just want to add to what Alexis said about the nitrogen. The nitrogen profile is an extremely important profile particularly when you're trying to work out how much nitrogen you require. And one of the components which has never been tested is the organic fraction. And it is the organic fraction in amino acids. And it's not only the amino acids, it provides the total amino acids, which is termed the water extractable organic nitrogen. And that is then with a series of, a sequence of analysis and formula, you can decide which of the available organic nitrogen will be available to the plants, and which will be retained by the soil and the soil system to retain or to feed your microbes. The more balanced you become, the more you release a complete amount of what's available, because the system starts equilibrating itself. So that is and sometimes that could be quite a substantial quantity of nitrogen, which has never been taken into account in previous nitrogen analysis, and so much so that we find that an over-fertilization of nitrogen has become an extremely common thing. And the over-fertilization of nitrogen creates just a myriad of other problems in agriculture.
Okay, that's great. Nicole, anything you'd like to add to that before we move on to the next question?
Well, I think there's really good research to show how your hot water nitrogen extraction aligns with hot water carbon. And so as we see those elements increase, you know, so your carbon's increasing as your nitrogen's increasing. So that's obviously going to have all sorts of other benefits apart from just nitrogen to the plant. But also seeing that's going to align with then our water holding capacity and water cycles. So we never talk about any of these elements in isolation because they are all linked together. So the minute we start to talk about microbial dynamics, how is that influencing the whole system? So we'll see improved, you know, water conditions in that soil or water availability to plants. So yeah, I think it's a really, really helpful test because there's so many different pools of carbon and nitrogen out there too. And I think this one gives us something that's more applicable in the field.
If I can just add something to that, when you look at a nitrogen profile, and the Haney test is a good example of how you can compare a nitrogen profile with the organic and inorganic components, because your total nitrogen that is available to soils are your nitrates, your ammonia, which are the inorganic component, and the organic component are your amino acid available and non-available. And when you look at a profile like that and you compare what we sample in nature and what we sample in intensively agriculturally chemically driven operation, you find a complete mirror image of one or the other. Nature's first call for nitrogen is amino acids, organic nitrogen. And in analysis where we look at nature, or a nature sample that's taken from an unworked virgin soil, you'll find that the amino acids are by far the highest quantity within that profile, and your nitrates are the least. And in a highly chemically fertilized environment, you find exactly the reverse. Your nitrates are by far the dominant, your ammonias are the least, and the organic nitrogens are a portion of the total. So it's a converse mirror image of one another, which just chose to indicate that the fact that we have 98% or 78% of nitrogen in the atmosphere is what nature uses, the natural systems to capture the nitrate, be it rhizobia assisted legumes or rhizobia free living rhizobia or other nitrogen fixing organisms that provide the nature given nitrogen in that system. So this is just to indicate that, as far as I'm concerned, plants prefer the nature-driven nitrogen from the atmosphere rather than the inorganic nitrates that's given as an external form of fertilizer.
Yeah, that's fantastic information, guys. Thanks so much for answering that question. So as we take as we talk about different soil health tests, let's just talk a little bit about, we talked about the Haney test and some things. Let's talk about the correlation between the Haney test and the PLFA test. Can you guys elaborate just a little bit on the correlation between the two, or when to one run, when to have one done in comparison to another?
Yeah, I started doing this work, and I did it with a lot of samples out of South Africa, and I added a number of samples that BuzzFeed sent to me. And what we did was we ran a correlation test between the soil health number. And this is another interesting thing about the Haney test, which we possibly just elaborate on. The soil health number is a formula that quantifies the three most important components in what I call the basic soil health reaction. The basic soil health reaction is the reaction that provides the food for the microbes, being the water extractable organic carbons, which are the sugars, the water extractable organic nitrogens, which we spoke about now quite extensively, and then the activity of the microbes that feed from the source, indicating what their total number within the system is, and that is the basic soil health indicator that drives the rest of the soil health functions, and it starts with actually with two processes, with a process of where organic matter is broken down, which is a catabolic process, and an organic process where the carbon is built. Once again, carbon from the atmosphere through a process of photosynthesis, exuded as sugars, and that provides the water extractable or organic carbons or the sugars that feed the microbes. So you have the water extractable organic carbon, water extractable organic nitrogen quantified, and the activity which is expressed in a 24-hour CO2 respiration. And those three elements are combined in the soil health number in the ANI test, which gives you an indication of how you stack up on the most important factor, or a little formula that drives all of soil health. And then just getting back, so what I did was I used that number, the soil health number or the soil health index, and I correlated it with the number of PLFA indicators, which were basically your total microbial biomass and soil organic matter, which is not part of the but it's also an important soil health test. Total fungal biomass, microbial active carbon, saprophytic fungi, and mycorrhizae. And we found some incredible correlations. Now, taking into account and understanding that the PLFA and the HANI are two separate tests. There is no way that any of these are used in the same parameter that will influence the correlation.
Oh,
Willie, we lost Willie, I think. He's frozen at the moment. So I guess, Alexis, Nicole, do you have anything you'd like to add to that?
Yeah, so I can kind of add to what I believe Willie was going to talk about, because I've seen this data. So Willie was very active before he was here. He was active in South Africa, getting samples from producers and everything like that. And so we actually ran the portions of the HANI test and the PLFA test on his soils, and found very strong correlations between the soil health score and the total living biomass. Now, Willie, you're back, so if you want to take back over, feel free.
Thanks, Alexis. Yeah, so we ran all of these, and you find incredibly good correlations. Like I'm just going to give you some of the actual figures, which is a combination of the South Carolina soils and the South African soils, and there's in excess of 200 data points represented in these correlations. We found soil organic matter correlates 0.83, microbial biomass 0.8, total fungal biomass 0.744, saprophytic fungi 0.72, mycorrhiza 0.73, protozoa 0.6. So, and obviously from there it decreases, which is an indication that the soil health number, although it is completely removed from all of these other tests, when you have a good soil health number, you are going to get good numbers coming out of your PLFA as a pretext to to your analysis. So and this for me is very, very important, which it actually provides more and more proof of the importance of the basic soil health reaction where water extractable organic carbon, water extractable organic nitrogen, and carbon dioxide evolution gives you a good indication of what it is the basis of your soil health.
Fantastic. So I do have, Nicole, is there anything you'd like to add to that before I move on to another question? Well, I think, so Willie, you talked about sugars and some things. So I'm going to segue into that. And so, Nicole, I'm going to direct this question at you and then see if the panel can't add some value there. Are all microbial amino acids considered amino sugars?
Yeah, the term amino sugars I've only been hearing in the last couple of years. And I guess anytime we're talking about sugars, we're talking about carbon, hydrogen, and oxygen. And amino acids have your nitrogen attached to that long chain carbon, hydrogen, and oxygen. So I guess you could consider them all as sugars. I mean, would you guys agree with that? Or, yeah, I mean, I think people are just coming up with new terminologies around these amino sugars as being the membrane of the outer membrane of microbial life, your bacteria, and really attributing that nitrogen cycle is, one of the main forms of nitrogen in the soil as being from the membranes of microbiology. So there's your amino sugars.
I think as far as the basic soil health test is concerned, you have to distinguish between sugars being the carbon and amino acids being the aminos, or the organic nitrogen. I haven't actually seen them being combined in any way. They could be combined in larger molecules, but as far as the Haney test is concerned, you have to look at them as two separate entities as far as microbial food. They are the two basic foods for microbes, and microbes prefer these in an organic form. Hence the sugars and the amino acids. The amino acids being the broken down from proteins through peptides to amino acids. And there's 28 known amino acids, but there's actually 54 amino-like type compounds which can be found. And the interesting thing is that as far as sugars are concerned, the reason for planting a variety or a mixture of cover crops is that each plant has its own fingerprint of root exudate that it will exude in the form of either sugars and or other substances which will attract a specific microbial biome to its rhizosphere, which it will feed. So the more diversity you have in your cover crops, the more diverse microbial biome you are going to create, which is going to support your soil function, maintenance, and improvement. Hence the ability to use your natural resources to better effect without having to use external resources like fertilizers and pesticides.
Nice. So I do have another question here, kind of staying on the topic of the PLFA, and forgive me if I don't say these terms correctly. So how does the PLFA differentiate between saprotroph and mycorrhizal fungi?
Yeah, so I can go ahead and take this one. So PLFAs are phospholipid fatty acids. So they are found in the outer membrane of bacteria and fungi. And they are very specific to genuses. So saprophytic fungi and mycorrhizal fungi have two totally separate fingerprints when we look at their PLFAs. So we have a formula that we utilize. We run all of our samples on a gas chromatograph, and then we take that data and we use our formula to populate what PLFAs actually fit what genus. So we can do that with not only saprophytic fungi, but mycorrhizal fungi, arbuscular mycorrhizal fungi, gram-positive and gram-negative bacteria. And we look for actinomyces and rhizobia in those categories also. So it's quite robust, our formula that we use. So yeah.
Wow,
It's
fantastic.
interesting because I was going to ask you that, because I was on the understanding that mycorrhizae didn't have the phospholipids, so they're not PLFA, but there's an NLFA, and that's how we test for. So you're doing both tests?
Yes.
Yeah, that's good. That's good to clarify that. Yeah, so I think it's more around the naming, whereas actually you're doing both tests.
Yeah, I think it's for me the overall value of a PLFA is to establish and quantify the presence of all your functional groups within the soil food web. Rhizobia is a bit of a difficult one because this one, the one and it's, correct me if I'm wrong, Alexis, the rhizobia that's tested in this specific PLFA are the free living rhizobias rather than the rhizobia associated legume species. So, and what I say to clients, because you rarely find a presence of them, but it's not only free living rhizobia that fix free living or that fix nitrogen in a non-associated legume way. There's several others from other bacteria from other species that are not legume related. So it's not if you get a zero rhizobia that you don't have any nitrogen fixing bacteria within your profile. But the important one is that you have a presence of at least some of each of these, and particularly the protozoa, which are one of the more important microbes within the food web that assist with nutrient cycling, and particularly nutrient cycling on predation on the predation level. That is obviously a topic of another discussion.
That's great. Anything else to add to that question, or you guys, can we move on to the next one? We're good. I'll tell you, you guys are doing a great job. It just blows my mind, some of the things that when we talk about soil health and regenerative agriculture and the way that we can truly impact the health of our soils and the dynamics of those kinds of things through some of these tests. It's just phenomenal. So leading back in, you know, the PLFA is getting a lot of attention right now, so that's very great. So PLFA would give a F and B ratio. Am I saying that right? Does that make sense? Would PLFA give an F and B ratio?
Yeah, that would be the
All right, help me out here, guys, just to make sure.
Yeah, so that's the fungi to bacteria ratio that we're looking at. And yes, our test does give that ratio for you. That's a pretty simple answer. So yeah, we give that straight to you on your report.
Fantastic. I don't know if you can see. I don't know if you can see the shade of red that my face turns when I have to read something that doesn't make a ton of sense to me.
Can I just say something on the fungal to bacterial ratio as it relates to PLFA?
Of course.
people always are looking for a one-to-one. If you get a one-to-one ratio on a PLFA, you are doing exceptionally well. For the simple reason that the concentration of fatty acids in bacteria are somewhat higher than what they are on a one-to-one ratio fungi to bacteria. So if you get a ratio of 0.25, that is more or less equivalent to the exact count, number for number, one-to-one fungi to bacteria. So anything better than 0.25 is a good ratio to have.
Nice. So let's, getting to some of the questions just regarding, basic soil health information in general. So hot topic, over the last couple of years is carbon levels and building carbon and the value of carbon in soil and how that correlates to soil health and regenerative agriculture. So question would be, how can I increase my soil organic carbon levels?
I'll start it off if you want. And Willie referred to, having diversity or having some of these diverse cover crop forage type species. So increasing the type of exudates that plants are sending down. And so you've got to consider there's kind of three pathways that are working in soil in terms of soil carbon. And what we're really wanting to do is build up that stable soil carbon component. So the component that's going to be there for longer. So I have the short cycle, which I call the I'm here for a good time, not a long time honey cycle. All that organic material that you're trampling or urine or plant roots or all that organic material, and it burns up, microbiology of breaking it down and respiring it off. And it's really important because it's going to help to stimulate plant growth. It's feeding microbiology. It's helping build structure. But when we're talking about, let's really get some carbon deeper down in the soil profile, then we are looking to those root exudates, which has been driven by photosynthesis, and laying down things like your chitin and your glomalin, like your different byproducts of fungal breakdown. And there's also a process where fungi will actually convert materials into crystals. So they might turn that into something like, say, a wedolite or a muluite, which are types of crystalline rocks or calcium oxides that are basically carbon that's in a very, very stable form. So when we look at our fungal to bacteria ratios, it is about how do I stimulate more fungi in the system, because they play such an important role in that drawdown of carbon and that long-term, more stable carbon. So we want that happening deeper down through that profile. And that's really, like a lot of people are measuring carbon, might only be doing that at 6 inches, and really, that's not how we assess how well is that system really regenerating. And what we're finding is people that are taking deeper carbon measurements are really seeing this buildup of not only organic carbon, but the inorganic carbon fraction.
Anybody else like to add to that?
Yeah, it's one of the best ways to sequester carbon is through photosynthesis. There's no question about it. Presently, I think we are losing about 11 gigatons of carbon at least no, not gigatons, pentatons, which is a massive amount of carbon per annum. And the only way that we are going to get back to that is through photosynthetic sequestration of carbon. So in anybody's lands where they farm, you want to have a net deposit of carbon rather than a return to the atmosphere of that carbon. And the only way you can do that is by keeping a living root with a photosynthetic capacity of leaves that will do that for you. And obviously the diversity, the better you're going to have that sequestration process take place. Because ultimately, when your fungi and particularly your mycorrhizal fungi, these guys can generate hyphae strands several thousand kilometers long, which is actually an extension of your root. And when they die, the debris is sometimes left in protected areas, in cavities, and this is also how your infiltration, water holding capacity, how your soil structure, how your aggregate stability, how all of that stuff improves. You get these hyphae wrapping around bits of organic matter, wrapping around bits of silt, bits of clay, and this is how you get these uneven and even and less even components that create the macropores where your oxygen and your water is stored, and this is the method that is also in the soil. If you can try and picture what's happening, because there's a million and one things happening at the same time. There's also other fungi and other bacteria that are excreting glues, like your mycorrhiza would be excreting glomalin, which is probably one of the most stable carbon components in nature, and that is a glue that sits there, and it is very calcitrant form of carbon, so it's not going to break down. So the more carbon dioxide that you can produce through your leaves, through your root exudates, feeding a growing population of microbes, and hopefully you're getting these to become stable and self-sufficient, self-sustaining, and ultimately providing a environment which will give you access to all the resources that's there, so that you don't have to use these from external purchased amounts, which is obviously will increases profit.
That's great. So there's a couple questions in the queue, but just generally speaking, so before I get to those that dive a little bit in more detail about some specifics, let's back it off just a notch, and let's say that I'm a traditional farmer, maybe traditional tillage or even strip till, right? I'm making the transition into less disturbance, but really just having an understanding is, so when I say, you know what, I want to test to see where my soil health is, and I run a Haney test. What does that data mean to me? What's the main points in there that I can say, oh, here's the number, here's the data set. I can then turn that into something else. Can you guys kind of just give us a general overview of, when I get those results back, what is the top three things that I provide? I mean, I know there's plenty, but what are the top three things that I should be looking for to be able to truly make an impact in the short term, immediate turnaround?
Okay. First of all, you'll have a reference point, because a soil analysis is like a balance sheet. It's a picture of that particular time and that particular day. An hour, 24 hours after you've taken it, you're going to see changes already in there, but you at least have a balance sheet or a data point that will reference your, give you an indication, as we said, on your soil health index or soil health number, that will be what you could use as a measurement or as your baseline against which you want to improve on. It will also give you a good indication of your nitrogen, and it will give you a nitrogen profile, which will give you your organic amount, which and the available. So you can measure that against what you want to achieve crop wise, and it will also give you the one thing which we did not touch on in the Haney is almost the second version, or the second edition of the Haney test, which is the H3A. It is, and just very briefly for our listeners, it is a root exudate simulated extract that Rick has has developed in order to simulate the soil conditions rather than using the harsh extracts which the chemical guys are attuned to. Obviously with the parameters which they use once they have those extracted to do their soil health amendments with. The H3A obviously is a mild oxalic citric acid type of extract, and it provides what is available in the soil system to you. So you will have that which is a more valuable set of information points rather than, as far as I'm concerned, than the harsh extract which you then have to use against set figures that have been detained over the years for this amount of zinc, for this amount of magnesium. This is what you apply. In the Haney, it gives you exactly what you have available in your soil for plant growth. Okay. So I think there's three important things. It's your nutrients, your nitrogen profile, and your soil health index. In other words, your snapshot of that moment in time against which is your baseline.
Great, thanks, Willie. Anybody else have anything you want to add to that?
Another great thing about the Haney test is that if you are a traditional farmer and you're used to using standard chemical tests, you're, like we said, you're getting half of your nitrogen measured. So it actually takes that and it compares it to the Haney test recommendation and gives you the amount of money you could be saving per acre. So to me, that's a huge figure that you can use and really as a selling point to traditional farmers.
Yeah, that's fantastic. So return on investment, essentially, it helps people to understand that you don't have to be completely regenerative or no-till to be able to take advantage of soil health analysis and to be able to kind of know where to start. And to your point, Willie, it's a good place to start, right? You got to have a baseline. So that's I appreciate you guys bringing some context to that. So we've got a couple more questions here that are in a little bit more detail. So again, bear with me on some of the terms here, but for good photosynthesis, the plant has to have access to some minerals. In case of poor soil biology, the inoculation of the seed efficient to bring back some bacteria, which have the capacity to, doesn't have the capacity to bring back some bacteria, which have the capacity to chalates, is that right?
Chelate.
There you go. That's why
you'll be done by the end.
Chelate some micro minerals. Did that come across? Was that too choppy? Do you guys understand the question?
Yeah.
Fire away.
Yeah, so it is, this is something that we would do on a basis during our transition is seed treatments. And I really think it's one of the most valuable things you can do because we're not putting a lot of a compost extract or a worm tea extract with some kind of trace element brew, so chelated trace elements. And when we're putting stuff on, I don't understand ounces. I'm sorry, you people live in this weird world. But like we're putting on, say, like less than a teaspoon of trace element per acre, if that makes sense. So we might chelate some of these trace elements and then use them as a seed treatment. And all it needs is like parts per billion as a catalyst really to enable that plant to really have the nutrients that it needs and then set that biology up. Because those PLFAs will show you there's a lot of biology out there. It's just who's active and who's not. And I guess it comes back to that question before around carbon. You know, what are you doing to kill some of these organisms? And perhaps you got to look at what's happening in terms of, are you putting a fungicide down with that seed, or what is your pre or post planting behaviors? You know, are you putting a soluble phosphate down? You know, all of that's going to have implications in terms of your whole microbial community and access to trace elements if you're doing these things to disturb how a plant would naturally take up elements.
if I can just add to that, Nicole, there's a, I don't want to call it a new biology, but it's quite established, an established method which is used extensively. It actually started in Australia by the use of autoinducers, extracting autoinducers and using them as a seed treatment.
Can you say that again? Sorry, you're extracting
Autoinducers.
oh, autoinducers, yeah.
Autoinducers, which are the chemical signal molecule that is signaled by the microbes that we are here. You must give us some food. And I'm just thinking. I think it's the Haggertys in Australia are using that extensively, and they are getting incredibly good results on their sandy soil. So that is something which one.
So what I'd like to say about that is that this is not new stuff. A lot of it, if you look at Korean natural farming or some of the indigenous methodologies of extracting from biology. So the Haggertys really are using, worm extract and compost extracts. The autoinducers are just part of, the secondary metabolites that biology are producing. And so very little, and then this comes to our quorum sensing or quorum signaling, very little is needed in these processes to set off germination or inhibit germination or suppress diseases or whatever it is. And I think it's so interesting to me that we see a lot of this as new when like so much of it's really old and so much of it is indigenous knowledge that I think needs to be acknowledged.
Great answer, guys. Don't feel bad about the ounces thing, Nicole. We drove to Minnesota with Willie, and we got lost 14 times because we kept trying to figure out what kilometers were in comparison to miles. So we've lived that reality too. So Willie's GPS in the back seat was going off, and we weren't sure where to turn. But got another couple questions for you guys. Do plants stop or slow down exudation when they reach their reproductive phase, or do they just change the makeup?
So that'll depend if you've got perennials or annuals, and if they're grasses or forbs, and if they are highly hybridized. Talking about some of the common crop cultivars have actually changed their ability to provide exudates to the soil. But if we're thinking about perennials, when you really think when they're going to that reproductive phase, they are going to be sending their sugars upwards and their nutrition upwards to that seed set. They don't need to be putting down roots or feeding more microbiology if they have what they need. And then you think, your forbs have this massive storage unit. So think about that too. If you're measuring bricks or photosynthesis, you're going to get a false positive high on annuals towards the end of the season because they're pushing their sugars upwards instead of a perennial, which is going to be really preparing itself for the long term. And when you talk about exudates, they could be all sorts of things. Like you think your knapweeds are pushing a whole lot of allelopathic type of exudates to stop competition when it's dropping seed. It doesn't want other things competing with it. So there's lots and lots of ways that those plants are using exudates, and it'll change from temperature, time of day, stresses. There's a constant flux happening with exudates.
Fantastic. Anybody have anything to add to that?
This was definitely a cycle of high root exudate exuding periods and lower exudate periods. And an interesting thing on that topic is that as soon as your particularly some of the annuals, when they start losing their exuding capacity because of lower photosynthetic capacity, you'll find that is also the time when your mycorrhiza starts making spores, because it is sensing that the mother plant from which they are living is starting to go into a senescent phase, and therefore they start sporulating, and that would be the high sporulating period for for mycorrhiza to establish their spores for that would be waiting for the crop to come back next season.
Fantastic. Well, we've got a couple more questions before we wrap up here, so I want to make sure we get these in. This question's pretty broad, so I'm sure we can go a ways on it. But do you feel the Haney can be used to make fertility decisions? Do you include recommendations with your soil health assessment?
Yes, we can, sure. I'm sorry, Alexis.
No, you're fine. I'm sure we were going to say the same thing. So yeah, we do include recommendations on our Haney tests. And I wholeheartedly believe that this can be used for fertility decisions because the H3A extract is like root exudates. And so you're really getting what's biologically available. And so you're really understanding what's actually in your soil and what the plants can use.
Fantastic. Anybody have anything you want to add to that? All right. Well, we've got one more question in the queue here, and I'll go ahead and get that. And again, please correct me at any moment. In doing homeopathic quantity or chelated traces, are you activating the dormant microbes that fix the solubilize those same traces, for example, molybdenum?
Yeah, so it's not quite homeopathic. So I wouldn't call it homeopathic quantities, like it is measurable. And yes, we have not done the testing in terms of do we see more molybdenum solubilizing organisms, but what we do see is the increased uptake, and we measure it in our leaf tissue tests of those trace elements beyond what we would have applied. And certainly when we've been making compost or vermicast, we add on small amounts of, what we do is try and match it to what is it that my soil needs. So say your soil is very critically low in phosphorus, of putting some kind of phosphorus, say like a rock phosphate or a guano, into that compost at pretty small amounts. But in doing that, we are breeding up those phosphate solubilizing bacteria, and we see a response when we put that compost out in terms of you're putting those organisms that are going to be able to release your bound nutrients. So I think that's a big part of the success of putting these small amounts in, is we're just supporting that workforce and basically just switching them on to do their work.
I can just come in and add to what Nicole said. In a completely regenerative situation, you'll find what I call a connectivity, and complete regenerative status is only reached when you have a complete connectivity between the plant root, the microbial, and particularly your mycorrhizal connections between the root and these hyphae being attached to mineral components. Then you'll find that signaling up and down takes place, and the plant will actually signal to the mycorrhizae and its helper bacteria what it requires. And these guys will then, through a process of quorum sensing, they will be provided enough nutrients to get to a state where there's enough of them to go over and do the job. They will then solubilize what's required, provide that to the mycorrhiza, and the mycorrhiza will then feed that within its tubes back to the plant. And once it has reached its efficiency, the food source to that particular combination of microbes will be cut off, and then you will have a predation process, or they will be predated and brought into as they will be predated, and their body parts or their nutrients will be exuded, into the soil system, available for other microbes or for plant growth.
Fantastic. Well, thank you for that. So I guess, you know, just in closing, I think we could talk about this topic for forever, right? I mean, there's so much information and so many relevant questions, but I think the beauty of it is there's so many different perspectives too, right? And I think that's what makes a panel like this so effective and so valuable. So I just want to take this time to thank each and every one of you guys for being on here. And you know, Nicole, thank you so much for your expertise. Willie and Alexis, thank you so much for joining us here. What I would ask, would you guys be comfortable putting your emails in the comment box? So if somebody has a question, they can reach out to you directly. If you're willing to do that, I think we could get a lot of value out of that, and certainly direct some of these additional questions that people may have in here. So if you're willing to do that, man, I'll tell you, I know that there's a lot of opportunity for these folks to be able to ask questions and continued education going further. So again, we will have a recording of this. We'll make sure that if you're interested in a link, we'll have some YouTube channels and different things like that. I have another question about the first Zoom meeting, so we'll make sure that those things get sent out to everybody. I'll put that in the comments as well, so that's out there. But again, Alexis, thank you so much. Nicole, thank you very much. We appreciate all your guys' expertise. Willie, thanks for joining and being a part of this for us, guys. And I'll be on for just a few more minutes, adding some comments in. But if you guys have any other questions, by all means, please shoot us an email. Go to our website, www.wardlab.com. Integrity Soils from Nicole. She's got some great resources out there. We're here to help. We're all here for the greater good. So please don't hesitate to reach out. Thanks, everybody. Appreciate the time, and have a great rest of your day.